In the second lecture of photosynthesis, let us try to understand the structure of the chloroplast, the pigments present in the chloroplast, and similarly the different types of chlorophylls that are involved in the photosynthesis process. Before going to the physiology and biochemistry of the photosynthetic process, it is very essential to know these structures. As I told you earlier, leaf, especially the chloroplast, the pigments, the chlorophyll molecules are very important. They serve almost as the platform for photosynthetic reactions. Now, physiology and biochemistry of photosynthesis. Then this is the overall reaction of photosynthesis. 6CO2, 6H2O give rise to a molecule of glucose, 6 molecules of water, 6 molecules of oxygen are released. The requirements are light, energy, electromagnetic radiation that comes from the sunlight, and these reactions take place in presence of chlorophyll. The chlorophyll is very essential. It is almost a platform for all photosynthetic reactions. Now again, this I want. Chlorophyll present in the leaf, in mesophyll cells, where the chloroplasts are present. Inside the chloroplast, the pigments and the chlorophyll molecule. The photosynthetic process takes place. CO2 from atmosphere, light energy, the light energy is required, and water is taken from the soil through the root system. Then glucose molecules, oxygen are released as the byproducts. Photosynthesis reactions take place in stages, not at a time, from carbon dioxide to carbohydrates. The reactions take place in a phase-wise manner because the first energy present in the electromagnetic radiation, that is sunlight, should be converted to chemical energy. That chemical energy present in the form of ATP should be converted to glucose for purpose of storage. Like that, the reactions take place in different stages. Capturing the radiant energy of sunlight by the chloroplast, one stage, first stage, capturing the radiant energy, sunlight, by the chloroplast, using the sunlight energy to make ATP, NADPH. ATP is energy currency, NADPH is reducing power in the light reactions or photochemical reactions. Once the light is absorbed, that will be trapped into ATP and the reducing power NADPH. These two together known as reducing power or assimilatory power. Using the ATP NADPH to power the synthesis of organic molecules from the atmospheric carbon dioxide during dark reactions or biochemical reactions. So thus 3 stages, these 2 stages in the light phase reactions, this is dark phase reactions, that is either Calvin cycle or C4 cycle. Carbon dioxide, which is in the oxidized state, is reduced to glucose, organic molecule. For that purpose, the assimilatory power is required. The assimilatory power is synthesized during the light phase reactions, photochemical reactions. So this is the structure of the typical chloroplast in eukaryotic cells, higher plants. A double membranous structure, two membranes. Then this inner substance is known as a stroma. This one is known as stroma. Inside the stroma, thylakoids or the thylakoids. At some regions these thylakoids are arranged just like sac-like structures. This is known as granum or grana. These individual structures are known as lamellae or thylakoids. They are also present in the stroma. These are called stroma thylakoids. These are grana thylakoids. Thylakoids, two types, present in the stroma region, present in the grana region. Grana thylakoids, stroma thylakoids. So sometimes they are also called, because they are arranged one above the other, they are called lamellae. In addition, here the chlorophyll molecules or pigment molecules are located, or all the substances, all the requirements that are required for photosynthetic reactions, light phase reactions are present in the thylakoids here. Whereas the dark phase reactions, Calvin cycle reactions, take place in the stroma. Besides the thylakoids, grana thylakoids, stroma thylakoids, there are present starch granules, drops of lipids. Then they also contain DNA. Chloroplasts have their own DNA. That DNA is double-stranded, circular, just like the DNA of the bacteria. This DNA. So therefore, these are autonomous cell structures. They can divide autonomously without much involvement of the nucleus. Then here and here, ribosomes are also present. DNA present, ribosomes are also present. Therefore, the chloroplasts can synthesize some of their own proteins, not all, but some of their own proteins. Thus, this is the structure of the typical chloroplast. Two membranes, outer membrane, inner membrane, then intermembrane space, and this is the stroma. Thylakoids, at certain places thylakoids are arranged or sacked one above the other. These regions are called grana. Single one is granum, grana. Then these thylakoids or grana thylakoids are interconnected by stroma thylakoids. These are stroma thylakoids. These are the regions where the active photochemical reactions take place. In other words, whatever the requirements are there for the photochemical reactions, they are located present here. In addition to this, they also contain the starch granules, lipid drops, then the chloroplasts contain their own double-stranded DNA. Circular double-stranded DNA, just like the bacteria. Then ribosomes are also present. Remember, these ribosomes are 70S type, not eukaryotic type, but prokaryotic type. 40S plus 30s, smaller subunit 30s, larger subunit 40s, together make 70s ribosomes. Then chloroplasts are present in photosynthetic protists and plants. Chloroplasts are variable in size, shape, number, but they share structural features, double membranous structures, inner structure, thylakoid structure, same. Possess chlorophyll and carotenoid pigments. Chlorophyll pigments, carotenoid pigments. Double membranous cell organelles. The inner matrix is called stroma. The fluid-like substance filled with the fluid-like substance, stroma. Stroma contains DNA, double-stranded circular DNA. Important. Double-stranded circular DNA. Ribosomes are also present. They are 70s, smaller subunit 30s, larger subunit 40s. Lipid droplets and starch granules are present. Stroma is the center for reaction of Calvin cycle reactions, dark phase reaction, whereas thylakoids are the centers for light phase reactions, photochemical reactions. Then again this. The most conspicuous structures of stroma are thylakoids, flattened membranous sacs. Flattened membranous sacs. Stalks of thylakoids are known as grana, singular granum. Grana thylakoids are connected by stroma thylakoids. Just now I have shown you the granum or sac-like structures, they are interconnected. Thylakoids contain chlorophyll pigments, carotenoids, electron carriers, ATP synthesis, because there the photochemical reactions, light phase reactions take place. Therefore, all the requirements are located there. What are they? Chlorophyll pigments, carotenoids, electron carriers, then ATP. Grana thylakoids are centers for light phase reactions of photosynthesis. Stroma, dark phase, Calvin cycle. Chloroplasts of some algae, chlorophyll members usually, they contain some pyrenoids. Then protein and pigments complexes of thylakoid membranes. If you take the thylakoids, grana thylakoids, stroma thylakoids, photosystem 1 or pigment system 1, pigment present in complex stroma thylakoids, PS1 in stroma thylakoids, PS2 in grana thylakoids. This point again important. Pigment system 1, where are they present? In stroma thylakoids, PS2 grana thylakoids. Then in cyclic photophosphorylation, B6AF complex will be there, that is evenly distributed both in grana thylakoids, stroma thylakoids. Light harvesting chlorophyll AB complex they contain, then ATP synthase complex in stroma thylakoids. Later we will study what is ATP synthase, that in stroma thylakoid. So stroma thylakoids contain ATP synthesis PS1, whereas grana thylakoids PS2. The first three, 1, 2, 3 are involved in light-driven electron transport and proton transport. Light phase reactions, first 1, 2, 3. The fourth one is light harvesting and has no photochemical activity. Only it harvests, it traps, it absorbs the light. This one. Then ATP synthase is involved in the synthesis of ATP. In light phase reactions, there is a synthesis of ATP. It is called photophosphorylation. Then this is the structure of thylakoid, grana thylakoid. The thylakoid membranous structure present in the granum region. The surrounding one is stroma. This is cavity. This in space. This space is called lumen of grana thylakoid. Then these are the membranes, this membrane and this membrane closed. Now various molecules are present, cytochrome B6F, then in the stroma ferridoxin, and here you see photosystem 2 with oxygen evolving complex. Photosystem 1 with NADP reductase. This photosystem mostly present in the stroma thylakoids, some are present even in the grana thylakoids also. Then this is plastoquinone membranous structure. These are all present in the membranes. And here this is ATP synthase molecules which catalyzes the synthesis of ATP from ADP and phosphate. This is called phosphorylation. This molecule, entire molecule is ATP synthase. Inside the lumen of granum, lumen means here this space, then the photolysis of water takes place. H2O, 2H2O undergoes splitting, hydrolysis, giving rise to 4 protons, 4 electrons and 1 molecule of oxygen. These protons are useful for building the proton motive force or electrochemical gradient, proton gradient. Because of this proton gradient, more number of protons this side and less number of protons, a gradient will be established across the membrane of thylakoid. This is responsible for synthesis of ATP because these protons try to go into the granum thylakoid, granum region, stroma region, and ATPs are synthesized. And this is a very informative diagram. The structure of one thylakoid with different types of molecules. Now, photosynthetic pigments, chlorophylls, carotenoids. Two types, broad category, chlorophylls A, B, C, D. In higher plants, chlorophyll A, B, C, but most common A, then B, C, D are less common. In bacteria, besides this, there will be a chlorophyll called bacteriochlorophyll. In bacteria only, not in higher plants. The chlorophyll that is present in the bacteria is known as bacteriochlorophyll. In plants, chlorophyll A, B are the main photosynthetic pigments. Chlorophyll molecules absorb light in the ranges of blue and red. The chlorophyll molecules absorb the light in the spectrum region of the light, where blue light 435 and red 680. Wavelengths and transmittance of green color, they absorb these lights and transmit, reflect the green light. That's why the leaves look green. These are the structures of chlorophyll A, chlorophyll B molecule. They are essentially similar. Central magnesium, this is a pyrrole molecule. This is phytol region, tail region, head and tail. The magnesium central magnesium is surrounded by 4 nitrogens here and here also. Phytol is also same, but only difference lies in this region. Chlorophyll A, chlorophyll B. Slight difference. This slight difference makes a lot of difference in their functions, performance. Structurally, chlorophyll molecules include hydrophobic, water-fearing tail. Let us go. This tail region is hydrophobic, water repulsive. Tail that inserts into the thylakoid membrane and a porphyrin ring head tetrapyrrole, because there will be 4 ring-like structures around the central magnesium, circular group of atoms surrounding the magnesium ion that absorbs the light. So here 2 regions, a porphyrin head, tail, that is a head and tail. This is hydrophobic, this is hydrophilic. Then carotenoids are another Cree group of pigments that absorb violet and blue-green. The light that is not absorbed by the chlorophyll A, B, they are absorbed by the carotenoid pigments, violet light and blue-green. Carotene pigment is orange colored, xanthophyll is yellow. Their functions help in the capture of light, as I told you, where the chlorophyll A, B are not absorbing, these pigments absorb those lights, which are violet and blue-green. Then another, they protect the chlorophyll molecules against the photooxidation due to excess of light energy. When there is excess of light energy, then the chlorophyll molecules undergo disintegration, they are destroyed. So these carotenoid pigments protect the chlorophyll against the excess light. That phenomena undergoing the disintegration is photooxidation. Against that, they protect the chlorophyll molecules. Photosynthesis 2 is located mostly in the grana thylakoids, whereas photosystem 1 and ATP synthase are mostly located in the stroma thylakoids and the outer layers of grana also, as I said earlier. Due to separate locations of the 2 photosystems, photosystem 1, photosystem 2, one here, another here, therefore there should be a system to transport the electrons or materials from photosystem 2 to photosystem 1. And that's why then some mobile electron carriers are present. Mobile, just like plastoquinone, just like cytochromes are required. They shuttle the electrons between them, photosystem 1 and these carriers are, what are they? Carriers of the electrons, plastoquinone, plastocyanin. Plastoquinone shuttles the electrons from photosystem 2 to cytochrome B6, whereas plastocyanin carries the electrons from the cytochrome F2 complex to photosystem 1. Later in the diagram I will show you. Plastoquinone from photosystem 2 to cytochromes, plastocyanin from cytochrome B6F2 photosystem 1. Together these proteins make use of light energy to drive the electron transport chains that generate chemiosmotic potential across the thylakoid membrane and NADH, the product of terminal redox reaction. The ATP synthase uses the chemiosmotic potential to make ATP during photophosphorylation. This point I will explain later on. In order to synthesize the ATP by the ATP synthase, the potential is required, chemoosmotic potential, or what is known as proton gradient, proton motive force. Then photosystem 1, two systems are there, photosystem 1, photosystem 2. What is the difference? Photolysis of water does not take place, no splitting of water, but there is a splitting of water. Here the reaction center is P700, here it is P680. It is rich in chlorophyll A, then chlorophyll B. But reverse here, rich in chlorophyll B, then chlorophyll A. Photosystem 1, chlorophyll A, chlorophyll B. Here photosystem 2, B, A. Molecular oxygen is not evolved because there is no photolysis of water. Here molecular oxygen is released. Receive the electrons from photosystem 2. The electrons that are required for the photosystem 1 come from photosystem 2. Here the received electrons from photolytic dissolution of water. Water undergoes splitting, as I said, 2H2O giving rise to 2 protons, 2 electrons and 1 molecule of oxygen. Those electrons are taken by the photosystem 2. Pigments absorb longer 680 nm wavelength light. Pigments absorb shorter, less than 680. Then in this reaction NADPH is formed. Here NADPH is not formed. It can participate in both cyclic and non-cyclic photophosphorylations. Just participate in cyclic non-cyclic photophosphorylation. The core complex is composed by less number of proteins, smaller number of proteins. The core complex RC reaction center consists of multi-subunit about 25 to 30 subunits. The photosystem lies on the outer surface of the thylakoid membrane, lies on the inner surface of the thylakoid. Thylakoid, two membranous structure, double membranous structure, outer, inner. Here on the outer surface, here in the PS1 have an iron sulfur type reaction center to quinone type of reaction center. The major function NADPH synthesis, reducing power. The main function is hydrolysis of water and ATP synthesis. So this is once again to you see. This is the lumen of the granothylakoid where photolysis of water takes place, and the outer membrane, inner membrane, the cytochrome for photosystem 1 and PS2 with oxygen evolving system, PS1 with an NADP reductase, ferredoxin, plastoquinone, plastocyanin. Look at it, this is present in the membrane, intermembrane protein, and this is inside the lumen where so here whatever the protons are released, they accumulate here. Because of that accumulation, the proton gradient is established, proton motive force or electrochemical gradient, and because consequence is that the protons are pushed out through the ATP synthesis. Because of this, the phosphorylation process, synthesis of ATP. Water photolysis. splitting of water, releasing oxygen. Hill's reaction. In 1937, Robert Hill demonstrated that isolated chloroplasts are isolated from the leaf, the mesophyll cells, can release oxygen in presence of light even in the absence of external electron acceptor like iron salts. When illuminated, the chloroplasts can release the oxygen. In other words, the photolysis of water takes place. External electron acceptors are called Hill's accidents. For example, NADP is Hill's oxidant. Under natural conditions, NADP acts as electron acceptor. So remember here, NADP is Hill's oxidant in higher plant. It accepts the electrons released from the water during photolysis. Rubin and Kamen proved that oxygen released in photosynthesis is derived from water, not from carbon dioxide. So this is the end of the structure of the chloroplasts, the pigments, the chlorophyll molecules, PS1, PS2, the ultrastructure of the thylakoids, the location of the different molecules required for the photochemical reactions, that is light chemical reactions. Then the differences between PS1, PS2, their locations. So in the next lecture we will see the light. What type of light is required for photosynthesis in higher plants? These aspects we will discuss in the next lecture. Thank you.